Hello, General Biology, and welcome to our fourth screencast for Section 1.3 from Chapter 1. And we were talking about ecology and evolution, and we're going to look at ecology and evolution of infectious diseases. Because there is a relationship that exists between hosts and the pathogens, and pathogens are the things that cause the disease. And what we're finding is they're constantly changing. Let me give you an example of how they're changing. So you heard of the disease called AIDS. Okay, AIDS is the acquired immune deficiency syndrome. Okay, so what happens is it's a virus, first of all. It's not anything that is living. So as a virus, from what we learned earlier in the chapter, a virus needs to attach to a healthy cell and inject its genetic information. So let's say that an AIDS virus attaches to Sally's cell. Okay, so let's say that the virus attaches to Sally's healthy helper T cells. So when the virus injects its nucleic acid, it's going to copy Sally's genetic code. So now if Sally, let's say Sally has a relationship with Bob. Okay, there's Bob. Now this virus, it's going to have a piece of Sally. It's going to get into Bob. It's going to reproduce in Bob. And now that nucleic acid that's in that virus is going to have a copy of Bob and Sally's genetic information. So let's say Bob's not faithful and has a relationship with Bertha. Okay. Now Bertha is going to get that virus from Bob, which has a piece of Sally's genetic information. Are you getting the picture? And let's say Bertha, doesn't like Bob, and she has a relationship with Ethelbert. And now Ethelbert's going to have the virus that has a part of Bertha, has a part of Bob, and a part of Sally's genetic information. So when you're considering how are you going to treat this, it's kind of hard to treat a virus that's going to change a little bit from one person to the next. So organisms that cause human diseases have their own ecology. That involves our bodies, the medicines that we take, our interactions with each other in the environment, and understanding these interactions and understanding cycles like this is crucial to safeguarding our future. So the example your book gives is a wildlife biologist will study a group of wild baboons, Gelder baboons, and they'll look at the pathogens that are in these wild animal populations and see how they may evolve to infect humans. We didn't realize it, but polio, which is a human disease, can be given to chimps. And there are diseases like AIDS, we'll go back to that AIDS, that came from the green monkey, that can be passed on to humans. So it's really important that we understand these relationships so that we can know how to treat them. One of the rising studies is genomics and molecular biology. That's understanding DNA, the molecules inside the cells, and we look at entire sets of DNA and how they code a wide range of organisms, and we look at the similarities within that genetic code. We're able to use computers that enable us to analyze databases of genetic information. So you know the size of your of a biology book. Your genetic information in your body would fill up 300 biology books. That's how much genetic information you have inside of you. So that's a lot of information, and we can put that into computer databases now so that we can understand mysteries of growth and development and aging and cancer and the history of life. We now know every chromosome in your body, where every gene is located, and by being able to understand that, we're going to be able to understand how to cure a lot of diseases and prevent genetic defects from happening. So in the next section, we're going to look at biological investigations. We'll consider the metric system. Science uses the metric system, so will we. The metric system is considered the universal scientific language because it measures things in tens and units of 10, and it makes it equal from one continent to the next when we use the same type of system and measurement. So we're going to be measuring temperature in degrees Celsius. We do it in Fahrenheit here, but we'll be doing degrees Celsius. We will be measuring length, so this is temperature. We will be measuring length in meters. And we know we can break down length into millimeters and centimeters so that they can be smaller. We're going to be doing volume in either liters if it's a liquid or cubic millimeters, so either or. This is a liquid, this is a solid for volume. Okay, so these are some of the common measurements that we're going to be based on. And they work out really well because they're physical standards that are scaled on multiples of 10. So it makes it easier to measure. Oh, I forgot mass. So here we go. I would like you to familiarize yourself with this data table. I knew I was leaving something out. So length, as we said, is going to be in meters. Mass is going to be in grams. Volume is going to be liters or cubic centimeters, as I said. And temperature, we're going to look at degrees Celsius. So if I was asking a question on the test, I might say the fish weighed 1,000, or let's say the fish weighed 5 kilograms. I might ask you, am I measuring length, mass, volume, or temperature of the fish? And you'd have to tell me mass. Okay? All right. So as we said, the basic unit of length is the meter. It can be multiplied or divided to measure objects of distance, large and small. We're going to be working with millimeters and centimeters most of all. And ladies, I need you to know on the ruler that the tiny little lines here are your millimeters and your larger lines are your centimeters. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is going to be your millimeters. This is going to be your centimeters. Okay? Get used to that because I'm going to be asking you to measure in millimeters and centimeters when you measure. Always worry about safety in the laboratory. Scientists that work in the laboratory or in the field are trained to use safe procedures when carrying out the investigations. So whenever you work in the biology laboratory, you must follow all safe practices. Before you start each activity, you're going to have to read all the steps and make sure that you understand the entire procedure, including any safety precautions. So you will be able to access on Readiker the labs before you do the lab, so it's going to be your job to read over the labs before you come into the classroom. The single most important safety rule is to always follow your teacher's instructions. So make sure you follow what I tell you. Sometimes I might amend the activity a little bit, and I will give specific safety precautions when those amendments are done so that you need to follow them. If you ever have any questions you're not sure, it's always best to ask me before you ask somebody else, because I know what I want. Because you may come in contact with organisms you cannot see, you need to make sure that you wash your hands thoroughly after every scientific activity. You need to wear appropriate protective gear, and I will tell you when you need your lab jackets or lab shirts and when you need your goggles to wear. You are responsible for your own safety, and you are responsible for my safety as well as your fellow classmates. So if we're not going to really be handling live animals, but we will be handling live microscopic organisms, and you're responsible for their safety as well. Okay, that ends the chapter. So you know what that means, test. We'll go over some things in class and be ready for the test. Have a good one.